Our next speaker is going to be from, that's going to be you? Okay. Is Diane Lee here? It's Diane Lee, it's not you.
No, he didn't make it to the conference.
It's not you, it's her. It's her talk.
Oh, it's not you.
Sorry. All right, I was confused also. So our next speaker is going to be from Sushmita Roy's lab in Wisconsin Madison, Diane Lee. And she's going to talk us about non-negative matrix factorization of high-C data. Yeah, PowerPoint is better. Just say cancel. You can use this clip if you want. Thank you.
Hello, everyone. I know it's getting close to lunchtime, so thanks for sticking around. I'd like to thank the organizers for putting together this really informative session. So, excuse me. The long-range gene regulation is one of the critical factors in development and disease in the mammalian genome. And as we've heard in previous talks, these long-range regulations, or the mechanism that brings together these far apart regulatory elements, is governed by higher order organization of the genome at multiple scales. So we have territories in which chromosomes tend to hang out in the nucleus. We have compartments that divides transcriptionally active and accessible stretches of regions from those that are repressed and inaccessible. Then we have our crowd favorite, TADs and subTADs, these self-interacting insulated neighborhoods. And finally, we have chromatin loops that brings those distant enhancer promoter elements together. And there are many existing computational methods that try to tease out the structural organization of chromosome, mostly at the TAD megabase scale. And they take very diverse algorithmic approaches. Some simply take an average across a sliding window in the high-C data. Others apply graph clustering. They also tackle the problem in different ways, or they look at the problem in different ways. 3DNet mod, for instance, is one of the methods that treats high-C like a graph and tries to find network modules. And then there are other methods that simply tries to find a boundary that separates the domains really well. So today, I'm going to add to the list. I'm going to present a method called Grinch that finds topological units of chromosome from high-C data. There are two key components of Grinch. The first one is non-negative matrix factorization, or NMF, and the second one is graph-regularization, which is a slight variation to NMF. So in the morning keynote speech, Cole Trapnell talked a little bit about dimensionality reduction techniques like TSNE and PCA. NMF is another one of those dimensionality reduction methods. It assumes that there's an underlying grouping structure in your data. So let's take the Netflix data challenge, for example. Here we have a user movie ratings matrix. This matrix might be millions of rows by millions of columns, but the idea is that there's a far fewer number of groupings of users, and similar movies. So NMF takes this large matrix and decomposes it into two factors much smaller in dimension. So here we have the movie matrix that recovers latent features about the movies, perhaps by genre. And then we have the user matrix, or user factor, that groups similar users with similar tastes and interests together. So the objective of NMF then is to minimize the difference between this original larger matrix and its approximation of it, which comes from the dot product of the two smaller lower dimensional factors. A lot of times you might have information about the existing relationship between the row entities or the column entities. So in our example, you might have information about the social network information about the users. And that might be helpful in grouping your users because people who tend to interact a lot more with each other on the social network actually might have similar interests and might want to watch similar movies. So in order to kind of incorporate this prior knowledge in a network format, you can apply graph regularization to NMF. The objective of NMF gets updated so that it adds two regularization terms. So here, we're going to regularize the user factor right there with the graph Laplacian of the social network whose information we're trying to incorporate. And the graph Laplacian is simply a matrix that captures the topology of the network whose information we're interested in. And you can do the same thing with the U factor or the movie factor as well. The idea here then becomes to create factors that is smooth on the graph topology. So taking advantage of the fact that Hi-C is already a graph that can be naturally represented as a matrix, we use NMF to find those hidden underlying grouping structures of genomic regions. We also take advantage of this distance dependency of interactions. Regions that tend to be close together tend to interact a lot more with each other. So we create this neighborhood graph and use it in graph regularization so that we influence the groupings of nearby regions together. So they're supposed to go together if they're nearby each other. So SPADIS actually penalize proximity. We're actually encouraging proximity when we're grouping things. So using that regularized factor, we actually we derive the final clusters of genomic regions that represents the organizational units of chromosomes. In order to kind of evaluate the biological significance of these Grinch clusters, we looked at the binding of CTCF, a very well-known boundary barrier or boundary element in the boundaries of the Grinch clusters themselves. So here you can visually inspect that the Grinch cluster boundaries align well with peaks of CTCF ChIP-seq binding signals. We quantified this enrichment using hypergeometric tests and measured the statistically significant fold enrichment. So Grinch here, oh, there's a pointer. Grinch here is represented in yellow. Levels above this red line right here represents above one-fold enrichment. And you can see that across 5 different cell lines, Grinch clusters are significantly enriched in CTCF signal at levels comparable to other graph-based methods like 3D-NetMod. You might notice that other more conventional methods like directionality, insulation, and Armitage, they recover boundaries, TADs whose boundaries are much more enriched in CTCF, which is an interesting observation. We also looked at other architectural proteins involved in chromatin looping, as well as chromatin remodeling and changing the accessibility of chromatin itself. And we see that again, Grinch is enriched in above one-fold in all of these different factors across 5 different cell lines, and at levels, again, comparable to graph-based methods like 3D-NetMod. We, again, notice the pattern that more conventional methods tend to recover much higher levels of enrichment boundaries, which is interesting. A lot of experimental Hi-C data, for those of you who worked with it, you might notice that they're very sparse and noisy. So in order to, we wanted to test if these different methods are stable to sparser input data or if their TAD results or their clustering results change drastically if you feed in a more sparser input data set. So we simulated sparser data by randomly setting X% of the original matrix elements to 0 as a dropout mechanism. And we also tried lowering the depth, overall depth of the data by scaling down every element by X% of its original value. So here in this chart, every data point represents the similarity between the clustering or TAD results that came from the original data sets versus the results that came from a sparsified version of the original data set. And it's measured by the RAND index, and RAND index of 1 being perfect agreement between those clustering results. You can see here in yellow, Grinch is one of the most stable methods amongst the methods that we evaluated. So Grinch yields clusters that are very similar to the original data set, even when it is fed a sparser data set of the original version. You might notice that methods like Armitage, 3D-NetMod, they actually failed to return any TADs as the level of sparsity increases, and for that reason, actually, installation score is not plotted here at all. When we look at lower depth data, again, Grinch is highly stable to lower depth data as well, at levels comparable to Armitage as well as 3D-NetMod. Armitage actually returned very small, tiny TADs, and then fewer and fewer of them as the depth got reduced. So if we take a look at kind of the sizes of these clusters or TADs that get returned by these different methods, this is a 2.5 megabase region and chromosome 9 of GM cell line. You see that Grinch returns much larger domains than some of the other methods that exist over here. And it's actually very similar to RGMap, which is another Gaussian mixture-based model, actually. And when we plot the distribution of TAD length, you again see that Grinch recovers a much diverse length of TADs over multi-megabase scale. So there's an interesting question of what is this functional unit that Grinch clusters represent, which is a question that we'll continue to address. One final property or benefit of using Grinch is that you can actually smooth out the original matrix by matrix completion process, or by basically multiplying back the original NMF factors. Again, this is beneficial because a lot of high-C data sets are sparse. So then you can take this smooth matrix and feed it into further downstream processing. For instance, something like FitIC, where you can identify regions that significantly interact with a SNP involved in a cancer, for instance. So, in conclusion, Grinch is a method to discover topological units of the chromosomes using matrix factorization and along with graph regularization. It finds clusters that are significantly enriched in different chromatin remodeling, chromatin looping elements. It is very stable to noisy and sparse data sets, and it can find TADs of diverse lengths. And finally, you can also use it to smooth input high-C matrix. Poster on Grinch will be on display this evening, so I hope to see some of you there. I'd like to thank Sushmita, who's my advisor, and Brittany, who'll be giving a talk later, and the rest of Team Roy back in Madison. I'd like to acknowledge the support and funding of NIH, as well as the Quantitative Biology Initiative. And finally, a shout out to our Center for High Throughput Computing. They provide the computers that we use every single day. So thank you for your attention. I hope you enjoy the rest of the conference.
I have two questions. So the first question is, NMF is known for being sort of unstable when you choose different numbers of components. Yeah. So I want to know how you address this issue.
Yes, so parameter selection is something that we're actively working on. Currently, we basically fix the K, or the cardinality, to basically half of the square root of the size of the matrix. That seems to give a pretty good range of different TAD sizes. However, another way in which you can pick those boundaries, pick those parameters, is if you have CTCF enrichment data available, you can do a parameter sweep and pick the best parameter combination that gives you the highest level of CTCF enrichment, because a lot of these structures correspond with CTCF peaks.
Okay, thank you. So the second question is, so you said that this method is very stable. I'm wondering if this is a good thing or bad thing. So for example, is it possible that it misses some changes, some subtle changes? So for example, if you have different conditions, maybe this is too stable that it can't detect the change.
That is a very good point, and that is something that we're actually working on actively. I'm trying to see if we can derive differential organizational structures from different clusters derived from 5 different cell lines, for instance. We do see a distinctive cluster boundaries, but we're not exactly sure how to interpret that and quantify it in a mass scale so that it can be useful as a biological insight.
Okay, thank you.
Thank you.
I would like to continue on the previous question. So is the stability of the number of domains and the relatively large size determined simply by the number by the parameter that you give to NMF?
Yes.
And so it's not really advantage of the method, it's just whatever you are giving to the program.
That is true, and I would actually say it is an advantage because you can change the number of clusters so that you can find a finer grained TADs or clusters or larger ones. Now, again, the challenge is interpreting the biological meaning of the smaller maybe loop structure that you got or a larger compartment level structure that you got. So it does give a flexibility. Again, the challenge is, what does it mean biologically? What do they represent functionally?
And again, the question is what parameter you give to NMF. And this is the only thing that determines the result.
Yes, and again, parameter selection can be done with CTCF boundary enrichment analysis or any other well-known architectural protein that you want to use. Thank you.
Thanks a lot. Sorry that we'll have to move on.